All right. So I also wanted to start with acknowledging the Aboriginal people and owners of the land we're meeting today and where we live. I think we are mostly visitors here. I mean, at least I am, but I'm really a happy visitor here because it's a great place to be. But it always has been and will be Aboriginal land, so if you can vote later this year, please vote yes. Thanks to Perry for organizing a wonderful conference. I'm really happy because some of my students, it's the first conference, and I don't think you can have a better conference as your first, because it goes beyond the science and considers much more than just impact papers and so on. So, and I think, I forgot his name, Honey? He gave a good talk, and it's really about values. And it's so important how we talk and what we do, and also how we talk with each other. And I think personally, I could probably chase what I did in my PhD and go after high impact journals and science and all this other stuff. But I think it's questionable at times working on those, because instead we can do really cool science, working on important problems for Australia, to everybody in our community and on our neighbors, of course. And I think the science we do is actually pretty nice, as you have seen these days. So I don't think there's a good excuse just chasing locked up impact factor journals. All right, that was my spiel. So now I give a talk, which is not really my talk. So really I got this talk from Rowan Kimber, from Sadi. I'm just basically giving this with slightly adaptations. Right, so it's really about a sentinel unit where we had a project on, a still ongoing project with University of Canberra, us at ANU, University of Sydney, then Sadi, and also data effects, and it was funded by DAF, by the Department of Agriculture, Fisheries and Forestry. All right, what was the aims of the studies is really, can you collect spores from the air and then extract them and detect myrtle rust? And can we also improve maybe the detection and also the methodologies around this and make it maybe potentially more cost effective in the long term? And I think it was also important just to show that we can collaborate across the whole spectrum of the sector or multiple sectors to generate impact. Right, so what's the project? So you might have heard of IMET-PEST. So IMET-PEST is basically a project which generated these air samplers. They're remote enabled, and they also have the solar panels. This is a different project, but that was basically a large project funded by multiple organizations, and we're using this here for myrtle rust, basically. So these spores are sampled, and then they're extracted. The DNA at Sadi, and we get some of this, and at Sadi they use some molecular techniques like qPCR to detect myrtle rust and all the other fungi basically which travel through the air. So this is a qPCR analysis, and what we at ANU added to this is metabarcoding. So qPCR is a method where you can detect one pathogen at a time, and metabarcoding allows you to detect multiple pathogens at a time in specific kingdoms or genera, depending on how you approach the problem. So this is the setup. You have this air samplers. It's Rowan who wrote this presentation. And that's one of these units inside. You can basically sample the spores, and you also have all the data like weather, wind, temperature, and so on. And that gets all integrated via data effects. And then these are placed out, and we have multiple sites. So I probably don't have to convince you that work on myrtle rust is important. It's a very interesting pathogen from basic science perspective, but of course it matters to really all of us. So previously, before the study, IMET-PEST had multiple of these sentinel units around the country, like as these dots. Here they were placed in the northern Queensland, here up north, but also some in Victoria and so on. In this panel on your left, I think it is, you can see then the detection of myrtle rust at different points in time. So you see like a spike here, spike here, spike here. That means myrtle rust was detected in spore samples at a given point in time with a qPCR assay, which like asks the question, is it there or not for a specific species? Then in the project, we have 3 sites. So one site was initially organized by Perry, you all know. And so this is here on the top, you see the temperature data, some weather data, and then the detection of myrtle rust with these peaks again by qPCR. So in the whole project, we're trying to look at 500 samples. The second site is now, or the site has basically moved into Royal Botanical Gardens. It's basically moved there in May, and there hasn't been much myrtle rust around, but it's probably just the weather. Here, another site is in South Australia, which is Mount Lofty Botanical Garden. There hasn't been any detection, but myrtle rust hasn't been really detected on, at least on plants, as far as I know, much in South Australia anyway. But there was a ping in the second site, which is at Adelaide Botanical Gardens at one point in time. But again, this is only the detection of spores, so we don't really know if it's actually infectious and has infected any of the Myrtaceae in the garden. So where do we come in? We come basically in with the metabarcoding. So instead of running qPCRs, we are asking the question, can we basically detect multiple pathogens at the same time? We focus on myrtle rust, but the idea is instead of running 50 qPCRs, you can maybe just do one sequencing run and maybe get similar answers. It's kind of here. What we are focusing on is fungi in the project. And so in fungi, we have this region which is fairly conserved. It doesn't work for all fungi, and there's like lots of caveats to it. But to keep it simple, you have these regions of the rDNA loci, which are ribosomal RNAs, which are important for translation. So you have this 18S and the 28S, and in between you have these regions which are called ITS. And they do vary between different fungi, so you can basically use the variation to call what fungus it is you have sequenced or where this read comes from. So we're using a technology which is called nanopore, and the main reason we do this because we can actually sequence the whole region in once, because it allows you to amplify sequence the whole amplicon, which is longer, so you can look at both regions at the same time, and that it's more broadly applicable in the sense, if we actually develop this technology, we can actually give it to people who can actually use it in their own laboratories or in field station because it's mobile, and instead of like 1.5 million, you spend $1,000 actually to set it up. So it's, I think it's quite exciting. All right, so we used different approaches. One of this was a student from computer science, so he trained like a computer science model, like machine learning models. And so what you see here is a confidence score, is basically how confident are we that a read which we know comes from Australopochynia is classified as Australopochynia. So we have a very high recall rate. And we do this in comparison with some of the other rusts, which Robert Park talked yesterday about. So we have like the stem rust causing fungus, stripe rust, and leaf rust. And all of those, we get quite good recalls and confidence scores. So we're quite confident that we can actually detect them with high confidence and recall. Right, so we also have been then working on data visualization, because this is how do we visualize how many pathogens and how much in there. This is an ongoing project. So this is basically read count in a certain confidence score where we of 0.9, so we have to be fairly confident that we detect that this read really belongs to that fungus. And you can see now instead of one fungus, you can actually detect multiple fungi at the same time, and you kind of can quantify it as well. And this one has some Australopochynia plus some other crop pathogen in there at the same time. So the idea is really to replace 50 qPCRs with a single assay. What's our future direction? So really we want to build a model where we can relate the qPCR quantification to the metabarcode outputs. So we do this over 500 samples, and these 500 samples will both have qPCR and metabarcoding results. So we can actually build models to predict the one from the other. And then we basically want to go beyond species identification. And this is a talk later, that's why I switched the talk, basically with some other people in the lab, because we want to go lineage calling. Because now we can look at the genetics of it with the sequencing, and not only the presence and absence, but what is actually there. And in the bigger picture, we would like in the future, maybe not for myrtle rust, but for other pathogens like stripe rust and so on, to look at virulence predictions, because you have the genetic information in your tube. Right. We're also working with the ACT Education Directorate and the Australian Botanic Garden currently to get one of these samplers actually to Canberra, and then in the term 4 to have like an educational program around this, how to use spore sampling and also insect sampling for conservation and biosecurity, where we hopefully get students to come to that in term 4. And of course, money makes the world go round, unfortunately, most of the time. So we hope to attract some more funding because I think it's, as you see, the room is full. There's lots of people working on this important disease. But I think we are like a little bit at a critical juncture if we actually attract more funding, at least for some of the more basic stuff we do. Right, so this collaborators, so we have University of Canberra, we have SARDI, ANU, us, University of Sydney Data Effects, and of course the funding from DAF. Thank you.
Thanks, Marge. It's a great talk and pretty exciting stuff. I was wondering, my question really was what was the air sampling rate of the system, and but with a view to understanding what the detection threshold would be of spores.
The sampling rate, and this is out of my expertise range, really. So this is really developed by Sadi and Rowan. So what we're currently sampling is the sample rate on a daily rate is basically sample daily, but I don't know how this gets sucked in and stuff. This is I'm not an engineer. For the detection limit, that's an interesting one. We actually have a project on this where we at least spike samples basically with a certain amount of DNA, and then we want to see how much can we detect with qPCR and how much can we detect with metabarcoding. I think we can potentially also do something similar with spiking spores in these samples and how many spores we need. So that's also still part of the project. Yeah. But we don't have a definitive answer. Yeah. The recall rate basically reflects the false negatives. So yeah, that's the false negative. The false positive rate is actually very much challenging, I think, because if you don't have because you never have all the species in the database, you never have all the species in your model, and this will also generate basically issues with classification to something which the read doesn't come from that species, but it maps to closely related species in your database. And this is a problem. We're trying to address this with some specific machine learning models, or basically higher dimensional representation of the data, which is similar to genetics, basically. It tries to capture the relatedness. So there's actually students working on this right now. But yes, but I'm actually more worried in general about false negatives for this kind of approach. Yeah. And I think it would also not replace qPCR. I think you would also have, hopefully, then you detect it and then you can follow up on it, basically.
My question is, what are you going to use the quantitative data for? Why do you want to know evidence?
I think the quantitative data, this was a biosecurity application of the system, but of course the system is also meant, some of it is actually applied for cropping systems, and there you actually want to know how much you have. So you want to know if you have like X amount of stripe rust around, or if it's just a little bit. So for these other applications, quantification is actually important. Maybe not that much in the biosecurity space, yeah.
So it might be more of a simple question, but with the devices, do you have to send somebody out to be collecting the data from the device, or is it able to be sent to you? And also, with that, is it, well, I guess more the first part of the question is more important, but does it have the capacity potentially in the future, do you see, to detect strains individually? Like we've talked about the dangerous strains in South America and elsewhere.
Right. So for the first question, so you can set the sampling rate of how long you want to sample at each of these valves, and the valves are on like a rotator, and so if you think it has 12 slots, depending what it is, what of the system it is, so you can basically have to go out there. If you do a daily sampling and you have 12 slots, you can go every 11th day or 12th day. And you could sample over longer periods too. The question is then how long these spores are viable and don't degrade and so on. But I think currently Perry in the project, I think she went once a week to the sampling site basically. And the second question, there will be a talk afterwards where we actually show that you can actually use certain methods even on the spore sampled material to potentially detect novel incursions. Yeah.